germline mutations in predisposition genes in pediatric cancer. Definition: recent progress in cancer genetics has identified specific loci that are involved in cancer progression, many of which have key roles in DNA repair, cell cycle control, and cell death pathways. Those loci, which are often mutated somatically during cancer progression, sometimes also contain inherent mutations. Recent genetic studies and quantitative population genetic analysis provide a framework for understanding the frequency of inherent mutations and the consequence of these mutations for increased predisposition to cancer.
Cancer is a group of over 100 diseases characterized by abnormal, uncontrolled cell growth. In a healthy body, cells grow, die, and are replaced in a very controlled way. Damage or change in the genetic material of cells by environmental or internal factors sometimes results in cells that do not die and continue to multiply until a mass of cancer cells or a tumor develops. Most cancer-related deaths are due to metastasis, malignant cells that penetrate into the circulatory system and establish colonies in other parts of the body. Great advancements have been made, but cancer is still the leading cause of death for people under the age of 85. In the United States, 1 in every 4 people die from cancer.
Applications in medicine. The frequency of germline mutations in cancer predisposition genes in children and adolescents with cancer and the implications of such mutations are largely unknown.
The human genome is a rich source of information. Clues to the causes and cures of childhood disease are buried inside, but analyzing the genome of a single human tumor yields an avalanche of data. And this is one of the biggest challenges for researchers, not just where to look for clues to disease, but how to look. Protein Paint is a new web tool created at St. Jude Children's Research Hospital to help scientists explore massive amounts of cancer data and make new discoveries. It includes tens of thousands of mutations for more than 1,000 pediatric cancer patients and growing, the largest database of its kind in the world. Here's how it works. Let's start by exploring brain tumors, ET. As we move outward, tumor types get more specific. We'll select an aggressive brain tumor type, high-grade glioma, or HGG. A cloud of different genes appears, all of which have mutations in this kind of tumor. Let's look more closely at this big one in the middle, a famous cancer gene called TP53. This long rectangle is a close-up of the protein made by TP53. Dotted along its length are mutations that occur in childhood cancer. But which ones are important? Let's use one mutation to dig into. The numbers show it's been discovered in 12 different patient samples, so it may be important. From here, we can dive deeply into a single patient sample. We see the mutation was found when the patient was diagnosed and other detailed information. We can even visit the journal article where the information was first published. In this way, we could explore all of the mutations found in this gene in different childhood cancers, or focus on a particular cancer type. With one click, we can compare genetic changes in a variety of adult cancers. This type of deep data helps scientists understand what is going wrong with specific genes in cancer, and may even help us design therapies that target those abnormal processes. Explore Protein Paint today at Pecan.
Pros and cons. The germline mutations identified in this study may provide insights into the causes of cancer. Knowledge of their presence may influence clinical management by directing cancer care, enabling presymptomatic genetic testing of relative kidney family planning measures, and facilitating the institution of potentially life-saving measures for cancer prevention and surveillance.
When a parent is first given that information that their child has cancer, one of the first questions that comes to mind is, why my child?
For many years, the scientific community reasoned that the genetic changes that cause pediatric cancer were simply unpredictable. Results of a new study by St. Jude Children's Research Hospital and Washington University, published in the New England Journal of Medicine, are changing the way we think about this question.
We pursued a study where we analyzed the genome, the normal tissue, from 1,120 pediatric cancer patients and asked, are there mutations in true cancer predisposition genes? And our finding was really very surprising. It was that 8.5% of children with pediatric cancer have a hereditary cancer predisposition. This really is a foundational study that raises the awareness to the field that there are patients that have germline mutations in their normal tissue. They're at increased risk of cancer, and we do need to start establishing exactly how to follow these patients. So at St. Jude, we have initiated a new study. It is called Genomes for Kids, and we are going to do an unparalleled level of sequencing on children with pediatric cancer.
By identifying which children have cancer predisposing conditions, we will be able to better treat these children, not only for the current cancer, but hopefully use the information to detect cancers earlier, later in our life.
With a commitment to rapidly apply these genetic research discoveries to patient care, St. Jude Children's Research Hospital has established a new hereditary cancer predisposition clinic.
We believe in working with the entire family because when you think about genetics and genetic conditions, they not only affect the child with cancer, but they have the potential to affect siblings and parents and other relatives.
The efforts made at St. Jude to not only understand the genetics of pediatric cancer, but to apply those findings directly to patient care have the potential to fundamentally change the way children from around the globe receive treatment.
In conclusion, the germline mutations identified may provide insights into the causes of cancer. Family history does not predict the presence of an underlying predisposition syndrome. In most patients, knowledge of their presence may influence clinical management.
Back in the day, as me and Mike Kugler's lab at Harvard, we were using some of his technology to develop the genome and look specifically at cancer genomes, find mutations. At that time, it actually turned out to be the most interesting stuff were the mutations we were finding healthy genomes. In fact, there were large deletion mutations, strange structural mutations in the genomes of everyone walking around on the planet. And that was a shocker that really didn't affect that much sort of variation in chromosomal structure as part of our naturally occurring integration. So that could have was really a big bomb went off in the United States that kind of highlights as an area that really had to be explored. It was obviously, genome-wide association studies were in full swing. Microtechnology was in its heyday, and you used those kinds of approaches to find deletion and duplication. And so this was an approach to go after, large populations, look for rare glitches in the genome, compare patients, help people to see if they have some rare glitches in the patient that you don't see in the general population. Sure enough, that's what we first, Mike and I started a project on autism in 2005, I think, and within a couple of years, we collected the data and to see quickly that there was a much higher frequency of rare glitches in autism compared to healthy systems. And just because of all there, as it turns out, this is not unique autism. In fact, the majority of severe brain disorders or psychiatric developmental disorders seem to have complex etiology, which include lots of rare. There's something about when you introduce a severe mutation into what happens is you have pretty significant effects on neurodevelopment. It can manifest as severe social impairment and learning disability in a child, or it can manifest later in adolescence. And begin to understand how the genetic glitches influence the brain, how the brain in a particular form of illness. That's not very well understood right now. But a basic sort of architecture of genetic architecture is a disease. Standard real itself. I think the common theme of the sort of
germline mutation are heritable changes in the DNA that occur in a sperm cell destined to become an egg or in the sperm. The zygote begins at the single stage. When transmitted to a child, the germline mutation is incorporated in every cell of their body. Germline mutations play a key role in genetic diseases. They play a role too in certain types of cancer death. For example, the eye tumor, retinoplasma, and womb tumor, and childhood malignancy of kidney. The germline mutation is confessed to those mutations like what?
Thank you.